Hello, my name is Professor Matthew Schmidt, and welcome to Genetics. In this session, we're going to continue our exploration of gene expression and protein synthesis, specifically talking today about RNA molecules and RNA processing. We did discuss last time the process of transcription, whereby an RNA molecule is copied from a DNA template. So the point of today's session is to look more at specifically what happens to the RNA under different circumstances, what needs to happen to it in order for it to be able to send its message in the case of messenger RNA, etc. So one thing that I think is not emphasized sometimes enough, and it has to do with the general idea of processing here, but I want you to understand something before we even look at the processing part. All messenger RNAs, and this is probably true of other types of RNAs as well, but let's focus on the message. Whether it's in bacteria, eukaryotes, whatever, when the whole entire RNA molecule is released away from the DNA, in other words, it's complete, it's finished, not only does it have a coding region, okay, in other words, the part that actually has the information that's going to go and get translated into a polypeptide, but it also has, so that's the coding region right there, but there's a 5' so-called UTR, or untranslated region, and that's located in this area, and then there's a 3' UTR also at the other end. So these untranslated regions, I mean, the name pretty much tells you what it is. They're untranslated, so they're not carrying coding information from the DNA, but they're doing something else, which we can discuss in a moment. Now, there is one difference, well, there are a lot of differences, but particularly in bacteria, once the messenger RNA, let's just assume we're talking about the message for a while now, it's mature and ready to be translated literally as soon as it's made. So in other words, this molecule that we're sort of diagrammed here, it's ready to go, it can interact with the ribosome and get translated, the part that's supposed to get translated. And remember, if you've been following along, we mentioned even that in bacteria, translation can start before transcription is even finished. These two processes are happening simultaneously in bacteria. In eukaryotes, on the other hand, the mRNA is immature when it's first transcribed and it's inside of the nucleus. So it's often referred to as a pre-mRNA during this time, and it's going to have to be processed in several ways before it can become a functional messenger RNA and leave the nucleus. So in the case of eukaryotes, for physical reasons, transcription and translation are separate, but also it's not just physical reasons. This mRNA is a lot different than the bacterial counterpart. Let's just go back for 1 minute to the bacterial one. The 5' UTR and the 3' UTR, we're not going to go into great detail about what's going on there, but it seems like the 5' untranslated region have something to do with control of translation. And remember, this is whether it's in bacteria or eukaryotes, which is why I just want to mention it very quickly. In other words, it may play a role in guiding messenger RNA towards a ribosome. We're not going to get into a lot of detail. The 3' region seems to have to do with mRNA stability, and these are very generalized observations. But my point is, the coding region obviously is the thing that's bringing the information from the gene to get translated. But there are regulatory aspects sort of built into this molecule. So how is it going to get translated? How long is it going to stay around? Things like that are inbuilt into the messenger RNA, okay? Prokaryotes or eukaryotes. Now, in eukaryotes, however, several things have to occur, must occur, before this pre-mRNA is mature and ready to go out to the ribosome and get translated. So let's take a look at a couple things that have to happen. First, at the 5' and the 3' end, at the very 5' end, excuse me, I wanted to do that, Capping has to occur. They call it a cap. So a modified G residue, which technically is called 7-methylguanosine. So it's a G, but this is a G with an addition that you won't see in any other place, has to be added to the 5' end of the RNA by a capping enzyme, and there have been several capping enzymes identified. But it's absolutely necessary for binding of the mRNA to the ribosome, and in fact, if there's any mutation that relevant capping enzymes, it's usually a lethal problem. This is also a little bit odd because we're adding something to a 5' end. Usually when we're polymerizing nucleotides, they're coming onto the 3' end, right? So for a couple of reasons, this is sort of different than what we've seen before. Also important is the addition to the 3' end of a so-called poly-A tail. So poly-A just means exactly what you would think. A lot of A or adenosine residues are going to get added on there. Anywhere between 50 and 250, average maybe 200. But so you get this long tail there. They're added to the end of the transcript. An enzyme called poly-A polymerase, that's pretty fitting, is what adds them. And this is very interesting. This is the first time we're seeing it. It can add these string of A's onto the end of a RNA without any template. In other words, you don't need a corresponding U to put it on. This enzyme has the ability to add A's over and over and over again. Now the tail functions in regulating the messenger RNA stability and transport from the nucleus. So we don't have to get into the details terribly, but don't get confused. Again, all messenger RNAs have the 5' and the 3' UTR, which certainly may have functions having to do with control and stability. Don't get that confused. In eukaryotes, the additional poly-A tail is put on there, and there's evidence that, obviously they wouldn't get put on for no function. So there's evidence that in eukaryotes, the poly-A tail, for example, is important in regulating the stability. Maybe the 3' UTR also functions in eukaryotes, but it's a separate thing. Okay. Now, in addition, and this is when we talk about RNA processing, there are a lot of things going on, but splicing is probably the most interesting, and most people probably think about splicing when they think of RNA processing. Now, this only occurs in eukaryotes as well. In the late 1970s, a major discovery was made, and the discovery specifically was this, that there was not, as had been assumed, strict collinearity. We use that term, and I'll make sure we understand it in a minute, between a mature, finished eukaryotic mRNA and the gene from which it was transcribed. So we've always said that, look, the sequence of DNA nucleotides, since the RNA is made from that, you would think the sequence of RNA nucleotides is collinear, one-to-one correlation between all of them, right? But it turns out that that is not the case with respect to eukaryotic mRNAs. So what you would see is something like this. If this was a mature mRNA in a eukaryote, they did experiments and they said, let's take the DNA template that this came from and try and hybridize them together. You might expect there to be just like this, right? Assuming that you could you can actually do this, get the single strand, break the DNA up into single strands and just use the single strand to hybridize. If there was perfect collinearity, you would see this type of a structure, and they actually looked under the electron microscope. Much to many people's surprise, what was seen was something more like this, with the DNA in blue. So there were parts that were collinear, but then you'd see these loops coming out. Okay? And the interpretation of the experiment was that these loops, obviously, so blue is DNA, red is RNA, that the DNA that was present in those loops, for whatever reason, did not make it. The sequence information in those loops did not make it into the mature messenger RNA. So what's now widely known and understood